Start recording. Okay. Okay, so in that case, I can kick off and get going. I mean, I hope this one will be perhaps a little shorter than the previous ones. There's not so much to say, really. It seems a more straightforward thing. So if I share my screen, we can get going. Okay, so is everybody seeing that okay? It's PowerPoint again. That working?
Yeah, it is.
Good, good. Okay, so this is the third part of this series on species distribution modeling, and about using species distribution modeling for forecasting. So just to recap on what we covered on the last two talks, the basic idea is we take the known distribution of a species, so the places where it's known to occur. We take maps of environmental variables like weather, soil, land cover, and so on. We try to form a model which compares the environmental conditions at the places where the species has been recorded to a random sample of the whole background and tries to spot a pattern in the places where the species occurs. We can then apply that model to the whole layers and get a potential distribution, which is the areas where the environmental conditions would seem to be right for that species, and therefore we might reasonably expect it to occur. Obviously, we can readily go on from there in that if we changed one of these layers, so for example, we substituted our weather layers with potential future weather from climate models, we can then get a predicted distribution under that climate change scenario. And this is something which you see being used quite a lot. So Roger and I went to International Symposium on the Surfidae in France last September, and there must have been 3 or 4 posters there presenting potential effects of climate change on the distribution of various species in various parts of the world. So it's a fairly popular thing to do. So what I want to do is run through the sort of availability of these climate prediction data for the UK, the means of actually doing this, and then have a look at some case studies and see what comes out of it. So, climate projections. Data is readily available. UK climate projections on a 5 km grid over the UK for the period 1980 to 2080. It was produced by the Met Office Hadley Centre, last updated in 2021. 3 time slices are readily available on this CEDA data archive for 80 to 2000, 21 to 2040, and 2060 to 2080. Now, I don't pretend to understand the climate modelling. There is lots of reference material available to describe the science behind this. There are various atmospheric models. This particular one is called GA705. The problem with all of these is there are lots of physical parameters which are involved in trying to predict what the climate is doing. You can imagine the model predicts the circulation of the air. How does that affect the transfer of heat to and from the oceans to the ground, and so on? and there's lots of stuff to do with the behavior of aerosols and the behavior of clouds and so on. So some of these variables are reasonably well understood, others are not well understood, and how they respond to changes in temperature is not necessarily well understood. And so the way they handle this is to have different versions of the model under different sets of assumptions. And the particular data we're going to look tonight looks at 12 out of 15 of these so-called perturbed physical parameters. There's also what scenario you model in terms of what's going to happen to carbon dioxide in the atmosphere. And the scenario I'm looking at is one called RCP 8.5. Now, if you go back to when the International Panel on Climate Change started, this was called the worst case scenario. As things went on, it was found that with each new IPCC report, what had actually happened was at least the worst case, and in many cases, worse than the worst case. So they started calling it business as usual, and it's these days called the business as usual model, which is basically what will happen if we just keep on emitting as we are doing at the moment, not change anything. And I think you'd probably all agree that that seems to be what's actually happening. So at the moment, this particular model by 2080 for the UK is suggesting winters will be warmer by 1.8 to 3.3 degrees C and wetter by 16 to 42%, and summers will be hotter by 3.6 to 5 degrees and drier by 16 to 46%. So we're seeing an increase in temperature. I think the central prediction for the UK is 3.8 degrees C by the end of the century. And we are seeing an increase in seasonality with wetter winters and drier summers, basically. And, you know, anecdotally, that's what we're experiencing. So fair enough. Now, if I just go and look at what this data looks like. So these are the files you can download from that data archive. They're in a format called .nc. You can see this is for precipitation, the daily maximum temperature and the daily minimum temperature. These are monthly figures for 5 kilometer squares, and they are for the set 01 of 15 for different sets of perturbations of these physical parameters. I've only gone for one of them. Ideally, I'd go for all of them and I'd run all of them and then do some sort of ensemble between them, but I haven't done that yet. So from these, we can extract monthly information, which is, so this is precipitation for January 1981, going up to, in the case of precipitation, up to December 2079 in that particular case. I then run these through producing bioclimatic variables for particular years. So if we look, for example, at 2021, these are the things like the rain in the coldest quarter, the total annual range of temperature, the mean annual temperature, the seasonality of temperature. These are variables that are reckoned to be biologically meaningful, which can be extracted from those precipitation, minimum and maximum temperature figures. So I've processed a series of these. These are 5 kilometer gridded figures for this particular, basically every 5 years through those 3 time slices, 80 to to 2000, 20 to 40, and 60 to 80. So these are what we're going to be using and going to be predicting on. So if we go back to this, the first case I wanted to have a look at was Leucozonia glaucia. And the reason for this is basically this dot map. This is a straightforward 10 kilometre square dot map in which the colours of the squares represent the latest year from, at which we have a record from that square. And what struck me about this particular map was this area of yellow squares down here. So the lighter colours are early, so yellow is before 2000. The red goes up to 2010, and this dark colour is post 2020. So most of the records down in the southeast corner of England are before 2000, or certainly before 2010. Yet this is by far the most recorded part of Britain. So does that mean we're tending to lose Leucozonia glaucia down in that corner? At the same time, if you look at this area up in northwest England and western Scotland, there's an awful lot of dark squares. So there are a lot of recent records there. Now, of course, we can't see under those squares, so we don't know what happened in the past on this particular map. So I generated this pair of maps. So this is just the 10 kilometre squares. We've got data from 1980 to 90, and in the last 5 years when I did this. These are deliberately very different length periods. They were chosen so that the number of records received by the Hoverfly Recording Scheme and the number of 10 kilometre squares from which data was received was pretty comparable. And if I remember rightly, the figures are something like 400,000 records and about 2,200 10 kilometre squares, give or take, in both of those periods. So they're quite comparable. And if we look at these two things, we will notice here that there is indeed this group of squares down in the southeast corner which are missing in this later map. And there is indeed quite a large scatter of records in this sort of area, which simply aren't there in this earlier map. And actually, it turns out our coverage of Scotland in this period was actually rather better than our coverage of Scotland more recently. So the fact they're missing there may have some significance. So what I did was I used Frescalo. You remember from the first talk I did on trends. This is a technique developed by Mark Hill, the previous head of the Biological Records Centre, to try and correct for recording effort in these sorts of records. So it tries to produce a relative frequency of species corrected for the amount of recording that's going on in the local area around each square, what he calls a neighbourhood of about 50 squares surrounding each of these squares. So if we look at the this rescaled frequency, so hopefully corrected for recording effort, and we compare what happened in 1980 to 90 with 2017 to 2021. What I'm showing here is the difference in this rescaled frequency between these two periods. If it's red, then it's increased. The rescaled frequency has gone up. If it's blue, then it's decreased. And if it's white, there's no change. It stayed the same. And pretty clearly, we've got a big blue area down in the southeast corner, which also extends through most of the lowland Midlands. And we've got red areas, increases, which really are in the high ground of central and northern Wales, the Pennines and North York Moors of northern England, and particularly in this area of northwest Scotland. And what that suggests to me is that Hoverfly Recording Scheme is telling us that this species is shifting northwestwards. It's declining in frequency down here and it's increasing in frequency up here. Now, if we go and look at the trend for this species, you'll see there was a drop through the 80s and 90s. Since, say, about 95, it's been fairly flat. There's no overall significant change. It's bounced up and down a bit. So there's no particular indication of a trend that the species has got a lot less frequent. Perhaps there was a bit of a fall in the start of the period, but since then it seems to have pretty flattened off, although it has jumped around quite a lot. And an interesting and quite simple result was if I simply take the centroids for these time periods, and that's take all the X coordinates from the records in that period and average them, take all the Y coordinates and average them, and what you end up with is an average coordinate of all the records in that period. And if you plot those centroids, there's a nice sort of northwest trending line. The distribution of this species seems to be moving northwestwards, whilst the overall frequency has not shown much sign of doing much. It's varied about a bit, but it's fairly flat. So it would be nice to investigate this species and see what the species distribution model will tell us. So you'll remember the last session we looked at how we could try and optimize a model to get the best fit. So I did that. I ran the stuff we did a fortnight ago, and I tuned the Maxent model, and I eliminated unnecessary environmental variables. And what I ended up with is a tuned model with these parameters, regularization of 4, factors of linear and hinge. This is a Maxent model we're looking at, and 500 iterations. And it's eliminated quite a lot of environmental variables, so we ended up with 13 out of 42. And if we take the test AUC after these steps, it was initially just under 0.8. By the time I tuned it, it was slightly over 0.8. And when I removed the unnecessary environmental variables, it was about 0.81. So if you remember the sort of rule of thumb stuff from the first session, 0.8 is a reasonably good model. It's nothing to write home about, but it's not bad. What can we forecast with that? So what I'll do here is I'll talk you through some R script to try and use those results from the climate modeling to actually fit a Maxent model and use them to forecast. So let's see. Okay, so we're going to use the data table package to handle these large data sets. Remember, our observations are about 1.5 million records, so it's quite big. We're going to use this package called Dismo, distribution modeling, to actually run our Maxent model. We'll use ggplot2 to plot maps and figures, and we'll use the Cairo package to produce nice output. And I'm just working my normal directory and clearing out variables before we start. I won't run all of this because it takes a fair amount of time towards the end, but we'll see what happens. So we're going to look at Leucozonoglossia. We're going to take a sample of 5,000 background squares. We're going to fit a model for the year 2021, and we're going to predict it for these years that I got those bioclimatic data from the Hadley Centre from the climate models. I just load here an outline of Britain, which we're going to use to produce maps, and define some functions. Now, R doesn't natively have a mode function, but you'll see in a moment we'll need it. This is just defining a mode function. You remember mode is the most frequent of amongst a set of variables, the one that occurs most often. Here's a function to load the environmental variables. And we've got to remember that these bioclimatic variables from the climate models are at 5 kilometers square distribution, whereas all our other data is at 1 km distribution. So if we start with the soil data, so this remember is the dominant soil type for 1 kilometer squares in Britain. If we load that, what we want is 5 kilometers. So what we've got to do is aggregate that to 5 kilometer squares. And this function, aggregate from the raster package, will do that. So it'll take this raster I've loaded. I'm applying a factor of 5. So instead of 1 by 1 squares, it's going to be 5 by 5 squares. And the function I'm using is the mode. So this is the dominant soil type for 1 kilometer squares. What I want is the dominant soil type for the 5 kilometer square. So I want the soil type that's most frequent in that 5 by 5 block, which is the mode. And of course, I want to leave out any missing values. Now, after I'd reduced the variables, these were the land cover variables that remained in: cropland, broadleaf, coniferous, mosaic tree and scrub, and urban. And if you look at, we looked at this, I think, last time, cropland was actually acting as a negative. If there was a high cropland, it was a good indication there wasn't Leucozona glossia there, whereas broadleaf and conifer were both positive and so on. Anyway, if we have a land cover for our particular year, we will use that year, but we've only got land cover data for 1992 to 2020. So if our year is outside that range, we just use the earliest land cover we got for 1992 for earlier years, and the latest land cover we've got, which is 2020 for later years. So if we're doing a model for 2080, we'll be using the land cover from 2020. That's the best I can do. I don't have land cover projections for 2080. Don't know if they exist. So we load that sequence of models. Again, we've got to aggregate them. Again, it's a factor of 5, so we're turning into 5 by 5 squares. But this is land cover. Land cover is number of hectares of that land type in each 1 kilometer square. So the function we want to apply this time is sum. We want the total amount of that land cover in that 5 by 5 square. Then finally, these are the bioclimatic variables that variable extraction found were relevant. And these, remember, are 5 kilometer square extractions. So all we need to do is load these. We don't need to aggregate them. So that will load our environmental layers. Finally, we've got a function to plot a map. So I think probably the thing to do is come back to this when we've got something to plot, so it makes more sense. So I'll just explain that a little later. So load our observations, extract the year, as we've seen all this before, clean up the grid references, get the 1 kilometer squares, load the coordinates for 1 kilometer squares and join them into the observations. And so we now should have observations with X and Y variables. I'm going to set a path to export my data. So let's just look where we're working. This is my hover stats variable. I'm in this thing here. I've got this thing called backcast, but I'm going to create a directory in here. So I generate the name of the directory, which is the name of the species, fitted, and the year I'm fitting to. So it'll be Leucozona glossia fitted 2021. If that directory doesn't exist, we create it. Now we'll fit our initial model. So let's just do that. The presences are the observations of that species from that particular year. Don't know quite why I've got that in twice. I don't need it in twice. Take that out. For that particular species, we're getting the XY coordinates, we're getting the unique values, and we're dropping any that are missing. So there are our presences. And in this particular case, there were 334 1 kilometre squares the species was present in. Then I'm going to get all the squares we got Hoverfly records from, and I'm allowing a 5 year, so plus or minus 2 years from our fitted year. That gives us 25,000 1 kilometre squares from which we had Hoverfly records, and we'll take a random sample of 5,000 of those. So there's our background sample of 5,000. Get the environmental variables for that year. This will take a little bit of time, and then we'll fit a Maxent model. So we pass it the environmental variables, the coordinates for the presences, the coordinates for the background, which factor which variables are factorial, and my way of indicating that is I start the name with CAT for categorical. And then I need to set arguments to apply that optimized model. And that's what these do. Now these are arguments to the Maxent. These are arguments to the Maxent function in Dismo. This is telling it to use linear and hinge factors, but not quadratic product and threshold, to use 500 iterations, and to use the regularization, which it calls the beta multiplier of 4, which were those factors we came up with by tuning the model. So let's just fit our initial model, which, remember, we're now running at 5 kilometre squares, so it doesn't take anywhere near as long as it did previously. It's a lot faster. We can have a look at what that produced. So we got a training AUC of 0.81. You can see the variable contributions there, and it used 249 presences and 5,037 background points. So what we're going to do now is loop through the years for which we have this bioclimatic data, this list of years here. And what we're going to do is get the environment for that particular year, predict using this model we've just fitted, plot a map, and if it's within the range for which we got Hoverfly data, we will test that predicted distribution against our real observations we got for that time period. So I will just, rather than make you sit through the entire loop, I will just run the first iteration of the loop. So I'll set I equals 1. So the year we're working with here is 1981. Just print it so we can see what we're doing. Get the environment layer for that particular year. Remember, our earliest land cover was 1992, so this is going to be using the land cover for 1992. Predict the accent model, predict the potential distribution using that accent model, and we can have a look at that. Which you can see looks pretty reasonable, really. And then we want to plot that map. Now, if I go back and show you how the plot map function works, we can now actually see it working. So we've got R, and I want... Okay. So I'm going to label this to use as a title with the year. So I'm just making a label from as character year. So that's pretty straightforward. Now I want to turn this potential distribution we produced into a data frame I can actually plot. So I tell it I want it to be a spatial pixels data frame, and then I want to convert that into an ordinary data frame. That should give us the probability of occurrence in that square, and then the XY coordinates, the square in this case for about 10,000 or so 5 kilometer squares. And I'm going to call the first one P, because that's better than layer, I think. Now, because of the way we're going to plot this, we're actually setting our breaks as the steps we want to plot in as 0 to 1 in steps of 0.2. This actually will use essentially it'll go from that value to just under that value. So if the value is actually 1, it won't be included in these steps. So what I'm saying here is if our predicted value is greater than 0.999, then make it 0.999, so it's just under 1. So it'll be in the top category of those colours. Right, now we're going to do a ggplot. So we call ggplot. I'm going to use a filled contour. So I'm giving it this XY data with 3 variables, the X coordinate, the Y coordinate, and Z is our predicted probability of occurrence. And I want it broken into 5 steps of 0.2 intervals. I'm then going to plot the outline, which is GB outline. So I give it the data outline again, XY. I want to color it gray, and I don't want it filled. And this scales the colors we're going to apply to these 5 values. And I'm using a function rainbow. I'm giving it 5 steps and telling it where I want to start, which is roughly red to light blue. Theme BW simply uses a plain white background, and coordinate equal tells it I want a 1-to-1 aspect ratio. My title, if you remember, is I've forgotten to use the label. I've just used year directly. Oh well. And then I'm removing the access text title, the grid, background grid, and I'm setting the title to be quite large fonts and telling it not to write a legend. So let's try that and have a look at it. So if I print that, So there we are, you see. So here's our year, 1981. We're showing the potential distribution as 5 steps of colour from light blue through green and yellow, and I think it's probably orange and red. Or it might be red and a much darker red. I can't exactly remember. But anyway, you can see the sort of impact that has. I'm going to print that to a file, so I generate the file name. And if we look at the file name, so this is in our path we want to use. I've called it Lucasona Glorsia 1981. That seems quite reasonable. And then I'm going to print that that file. So if we go back down here, we can see if I run plot map, that's what will happen. Right, now, if it's between 1980 and 2022, the period for which we got Hoverfly records, then I actually want to use the real observations we've got to test how good that predicted distribution is. We're in 1981, so it's greater than 1980, so that'll happen. So it'll get the presences for that particular year, and you can see there were 54 of them in this case. And then it'll evaluate the model using those presences, the background data we had before, the particular model, and the environment variables we loaded. And then I want to save some results. So I set up this blank data frame called result. And in each loop, I will add a new row to that result, which includes the year, the number of presences, the AUC from this evaluation, and the equal sensitivity, specificity threshold for that evaluation. So if I run that, we look at the results. Whoops, I obviously didn't run that command for some reason. Now it should work. You can see that got an AUC of 0.72. There were 54 presences for 1981. So if we run that, we will get a plot for each year, and we will accumulate the results. And what I'm doing here is plotting the year against that AUC, so I can see how well those different models fitted. Again, we're using ggplot, we're using the result. The X value is the year, the Y value is the AUC. I'm adding a red line through those points, and I'm plotting the points as grey shapes filled with red. I'm labeling the x-axis as year and the y-axis as AUC, and I'm increasing the size of the text on the axes a little bit. And if we look in our thing here, we'll see what that plot looks like. So here's AUC against year, and these are the values. These are the maps we produced. And finally, this results data frame I'm saving to a CSV file, so we can actually look at that data. So there's the results for that series of years. Okay, so that's what's involved in making this particular set of predictions. So let's go back into the PowerPoint and actually and have a look at what we get. So here's the results for Lucasona Glorsia with the model fitted in 2021 and then predicted to these various years according to the climate model. So there were 343 presences, as you saw. I fitted it to. The fitted AUC was just over 0.8. It generally has declined away from that as further you get from the fitted point, the worse it fits. We look at these pictures, we can see 1993 particularly looks a bit peculiar. And we've not really, in 1997, we saw this splodge in southeast England, but I mean, not really at all in 1985. 1985 fitted particularly badly. So I wouldn't say this has been massively successful at predicting going backwards, where we actually have Hoverfly data to compare it with. If we now look at the forward projections, these are the years between 2020 and 2040, and here are the years between 2060 and 2080. And you can see what it's predicting is this general north-westward shift in the distribution of Leucozona glaucia. And by about the mid-2070s and 2080, we're running out of land in the northwest, and so we are pushing this species out, essentially. So the forecast is suggesting this further trend of northwestward movement. I thought it'd be interesting to look at species we know something about how it's been changing. So I had to go with Polycella zanaria, and again, exactly the same as before. I tuned the model. I got pretty similar results, but now it's linear, quadratic, and product factors and 750 iterations. And very strangely, nearly all the environmental variables got eliminated. I only ended up with 4 environmental variables, and the test AUCs never quite got to 0.8. Although clearly they did improve with the tuning and the reduction in variables. So if we fit, in this case, I fitted for 1993. We think this species started taking off around about the mid-90s. So this is really before the big expansion of range. So it's this one here. I had to allow observations for the year before and the year after to get any reasonable number. So I got 75 observations to fit the thing. And as you can see, the fit of the model declined pretty precipitously from the fitted year. Interestingly, behind the fitted year, despite the fact these don't look good at all, the AUC stays relatively high. I'm not quite sure why that happened. But basically, you can see that 1993, we basically got it in the London basin. By 1999, it started to spread. And by 2021, it's quite widely spread. And if we look at the predictions here, for the 2020s, the 2040s, it's continued to expand northwards. And for the 2060s, the 2080s, really by the late 2070s, it's occupying pretty much all of Britain. We're getting records right up into, I think it even gets up into Shetland, maybe, certainly into Orkney. So I thought I'd try a different tack and actually fit the model for 2021 and back project as well as forward project. This time I could just use 2021, and we had 1,600 presences, so we had a lot of data to fit it to. So this is where it's fitted, but you can see if you back project, it's not really getting this fact that the range was much smaller back here at all, really. And interestingly, that the AUC is still going up and up and up. So again, not quite sure why that's happening. It's a bit strange. Only 1993 looks anything like convincing. And if we forward project this model, pattern's sort of similar, but nowhere near as frequent. In this case, it's still getting over pretty much the entire British Isles by 2080. So I thought it'd be useful just to have a look at a species where there doesn't seem to have been much distribution change. This is the overall 10 kilometre square dot map for Christchurch and Cortum. And this is the 1980 to 90 compared with the 2018 to 22. So the last 5 years of data at the current moment. And you can see distribution hasn't changed much. We get the odd outlier. We have actually checked this one as far as we can, and it seems to be okay. We did actually have a spot up in Kubrish, which turned out to be quite wrong. But anyway, and the trend is pretty flat. It went through a decline through the 1990s and early 2000s, but seems to have recovered from that. Again, if we tune the model, I got a regularization of 1, linear and hinge factors, 1500 iterations. Again, it got rid of most of the variables, only 7 left, and none of those were land cover variables, which surprised me a bit. So this is entirely soil and climatic variables we're working on. And if we, in this case, I fitted to 2021 with 79 observations, and I back projected. Doesn't look too bad for 1999 and possibly 1989, but what's happened here and these, they really have not worked very well at all. Again, AUC is doing funny things. And if we forward project, well, it's sort of all over the place. We get in 2025 it racing all the way up to northern Scotland, and in 2029 back to a bit more than its current range, and then sort of oscillating around. So in 2073 we've got it much less common but quite widespread, and so don't know what's going on here, but it's not behaved itself at all well. So what do we make of all this? So forecasting how distribution of species may change is obviously a pretty compelling idea. And the fact that we've got data available from these climatic models, potential future scenarios, it's very tempting to plug them into our species distribution model and see what happens. We have a big advantage with the Hoverfly Recording Scheme in that we're sitting on 40 years of data, which we can backcast over to test how well it's working. And I think you'll see from the examples I've shown you, the answer is really not phenomenally well, really. I've not felt the results are terribly impressive. The AUC tends to fall off from the further you move away from the time slice to which the model was fitted. And some of them behave very strangely, like Christchurch and Cortum. I get the impression from looking at the literature that machine learning models like Maxent do not predict terribly well in these scenarios, so they're probably not the best models to use. Regression-based models like general linear models or general additive models might be better in this respect. I'm afraid I haven't spent a lot of time trying to do that. But my general feeling about this is if you add the uncertainty of a species distribution model to the uncertainty of a climate model, it seems to me the results you're producing are pretty dodgy, really. And I certainly wouldn't want to be betting my mortgage on any of the results coming out of this. So I'm quite skeptical that this is really a terribly useful thing to do, really. And that's about what I wanted to say, really.
Oh, that's terrific, Stuart.
Yeah.
It leaves us really questioning an awful lot of things, doesn't it? The models are useful, but they are only models, and we shouldn't actually place too much faith on them, and be too certain about what they're producing.
My feeling is that something like Maxent is really pretty good at taking data which may be incomplete and trying to fill in the gaps. And it seems to do that reasonably well. But if you then start predicting that to other time slices, it pretty quickly breaks down. And we obviously, for the hoverflies, we're in a really privileged situation where we have quite a long run of data to test that on. And that is great. And many other people from many other groups don't have that. So perhaps we ought to be setting a good example.
Stuart,
yeah, the thing I would think, though, about, as I understood it, Maxent was designed not to forecast, but to actually do as you say, fill in gaps.
Yeah.
And therefore, one's trying to use Maxent for a purpose that it wasn't designed for.
Well, I don't know. I mean, Maxent, if you go back to where it came from, it's actually its roots are in machine translation and speech to writing, those sorts of purposes. That's where it came from. So a very different sort of field. I mean, it'd be interesting to have a go with some of the other modelling techniques.
Stuart, thanks very much for that presentation. It's great. I'm new to this group. My name's Nick Grattan, but really interesting. I wonder if there isn't a factor here of a dispersal rate, because the model here is almost saying that if there's a temperature change in a particular area, we almost immediately expect the surfer to be present in that area.
Yeah.
And it doesn't take into account, perhaps over the 3 or 4 years past, there has been a general dispersal rate. And I think if we were to build in some sort of memory into the model, and this is done in some of the machine learning models, it's long, short-term memory, that it actually can build up a knowledge of where it was and where it should be, and build in that time aspect, which is probably missing from Maxent at the moment.
Yeah, I should have said that with Volucella's and Aria particularly. Going right back to the first of these 3 talks, one of the things I talked about there was what these species distribution models were intended to try and predict is the potential distribution, which is places that seem to be suitable for the species. That doesn't mean they're actually occupied by the species, because there are other factors. And those can be things like physical barriers, or competing species, or human persecution, or whatever. And clearly, in the case with Volucella zanaria, as you say, the fact that the climate envelope is expanding may be happening faster than the species can actually keep up with. So you might well expect there to be a time lag while it manages to fill that potential climate envelope. And I should have said that, because it clearly is something that matters in a case like that. And there's been quite a lot of work on this sort of stuff, as you mentioned, because one of the things species distribution modelings have been used for quite a bit is where you get invasive species coming in, you want some idea of where those invasive species might go, and therefore you can try and tune your resources and your response to the most critical areas. and so they also contain an element of process modeling, which involves sort of population dynamics and the dispersal ability of the species and things.
Yeah, because you can have the reverse effect that if a species, if I can describe it as, cannot keep up with the change, it will become extinct. Yeah.
I mean, I've got a suspicion that with Chrysotoxinum quartum, that it's a species we've sort of been expecting to move. It's one of these quite thermophilic things that you might expect to be able to expand northwards as things become warmer. But we've seen no sign of that yet. Now, is it possibly because it can't for some reason? Maybe it's a poor disperser, maybe it finds it difficult to get from one habitat patch to another. Maybe it's a competition thing because you get this completely exclusive distribution with arcuatum. And there are very few 10 kilometre squares with both of those two species in. So maybe the fact that arcuatum is there keeps it out in some way. We don't know. So there may, as you say, well be other things going on apart from just climate warming.
Thank you.
Fine, last question. Well, first of all, I was going to say thanks, Stuart, for putting these on, because the initial one with the trends made R seem much more accessible than I thought, and I've in fact got it running on my machine now and starting to do things. So you've had an impact there.
Yeah.
So somewhat different. I'm not aiming to do anything as elaborate as you are here, but for instance, with things like the anti-Mayed recording scheme data, I'm more thinking that it's really only realistic to look at species distributions by vice county and adopt a much more broad brush approach.
Right.
But the question I was leading up to was that I used the term machine learning, because I did look at the reference you gave on the first one, Elith and co in 2010, and it talked about translating initial concepts or expressing it as a statistical method, which did make it sound much more like a fitting operation, but using entropy as or entropy difference as a measure of difference between the observations and the overall distribution of environmental variables. So is it really machine learning has connotations of artificial intelligence. Is it really part of that, which we're hearing a lot about in the last few days?
Well, I think machine learning is at its root a sort of feedback loop where you're taking the results, calculating some sort of difference from what you expect, and feeding that back into changing the parameters until you get closer to that expectation. And certainly things like Maxent or Random Forest are essentially doing that. It's a penalized learning model using a loss function, which in the case of Maxent, as you say, is an entropy difference.
It's just a matter of terminology, really. It gave a completely different impression.
I mean, clearly, if you're doing with an artificial neural network, then you're directly using the same sort of thing as deep learning models and so on.
Right, thanks.
I mean, I think part of it is you end up with a black box where you're not too sure what's actually going on inside. And it is quite difficult to actually poke into it and find out exactly what it's doing and how it's doing it. And that would certainly be true of most of the current AI stuff that has made the headlines and so on, where it's actually finding out how they're doing what they're doing is one of the big areas of study, and is quite tricky.
Well, so it's a change to see a presentation where the results of this model doesn't work very well for forecasting at all, because there is the publication bias, which probably still exists towards studies which do show some degree of success of the modelling. So no, whether this has implications for the wider applications of SDM would be interesting to speculate on.
Well, I'm not sure there are too many people who've actually got access to the sort of run of data that we have, who've actually been able to test whether their predictions actually work against real data to the same sort of extent. I mean, I'm sure it must have been done for vertebrates.
So many of those are quite difficult to actually survey very systematically. It's like the Mammal Society used to say, possibly you have got more data per species, just because there's far fewer number of species.
We do have a slight complication though with all of that. That whilst we have, if you look at the data sets that Stuart used, he was using a very long run of data for the 1980s. And a very short run of data for the last 5 years. And so one of the problems that we have, I think, is that the 1980s data are compiled by a slightly different recorder subset.
Yep.
And that might mean that actually one of the problems for Volucella zonaria was literally that there weren't enough boots on the ground to pick up the distribution as held at the time. Now, it may or may not be true, but the reality is that we've now got upwards of 3,000 people each year looking out for hoverflies, many of whom will particularly record Volucella zonaria. If you go back to 1983 or 4, there might have been 150 people, of which possibly 20 were really active. So that there are, I think that probably helps to explain why some of the hindcasting perhaps doesn't work as well as you might have hoped it does. I also wonder with the forecasting whether what we're looking at is that you are using averaged temperatures. And if I remember rightly, Stuart, when you did the model back in 2004 for Volucella zonaria, you picked up two critical parameters. One was the heat of the July maximum, and the January minimum. And it may be that we're picking up some other factors now that perhaps weren't as obvious as they were then.
The bioclimatic variables that came out, particularly, as I said, there were very few variables.
Yeah.
Volucella zonaria. They were the soil type, urban coverage, the temperature of the coolest quarter, and the temperature of the warmest quarter.
Right.
So, in fact, sorry, the temperature of the warmest month, not the warmest quarter.
Yeah.
So,
I guess urban coverage could be picking up heat island effects, could it? The fact that urban
but also
coverage,
the fact that probably most of the records the recording scheme gets tend to come from people's gardens.
But actually, when you're out in the field, my experience is that you don't see Volucella zonaria very often. You do see it often in urban situations. So there are potentially two factors. One is perhaps urban heat island effects, and the other is perhaps something to do with the circumstances under which the wasps whose nests they occupy are constructing their nests. It may be that there are more opportunities for certain nest types Mike might be in a better position to comment than I am, but ground nesters, I'm wondering, in urban situations, might actually have warmer ground nesting situations.
Right. I really want some olfactory response data here, because your distribution actually shadows the distribution of Vespula germanica and not vulgaris, which I quickly emailed to you.
Yeah.
And it may be that germanica, which itself likes a warmer climate, is the host and not vulgaris. The only person who might have information on that is a guy who came over looking for volucellas to control Vespula in New Zealand. And I have to go back and look at, see if I can find his name, some 10 years back. But he was actually taking volucella larvae and checking what they actually did.
Yeah. But he was doing volucella inanis, wasn't he?
He chose volucella inanis.
Oh, right. Okay.
He screened all the volucellas.
Oh, right. Okay.
How he knew that inanis was actually a predator, whereas the rest were more scavengers.
Yeah.
Well, that was exactly it. Yeah, it's just inanis is technically a predator. Yeah.
But I think he actually screened some of the others. So if I can find his contact from the old BWAS things, I think that's
it ought to be possible to find.
What's happened is that Germanica has now spread much, much further north.
Right.
Yeah. And that shadows, it's only correlative, but that shadows the Volucella.
That's interesting, isn't it?
That was immediate what hit me the minute that pictures came up. That is the same kind of trend, distributional trend, that we're getting with that Vespula.
That is interesting.
Yeah. Don't build too big a cast on it, but the answer would be: can we find out whether there are olfactory clues? Don't forget, these things work through olfactory more than visual.
Yeah.
Oh yeah, absolutely. 100%. We rather anthropomorphize a lot of what things are working on, but if you actually look at the range of chemosensors that they have, it's much more extensive than we might imagine. Of course, that raises the question. I mean, maybe there is something there. Can we get people to collect samples of wasps' nests and the Volucella larvae and see if we can actually get some decent data on that? I don't think
we will need samples, because those two wasps, although on an average you can deal with them in the field by the length of the stripe behind the gena, it's not absolute. So you'll need samples, multiple specimen samples, not just one.
Yeah.
Okay, if nobody's got anything else, there was one other point I would like to raise, if I may. I would quite like to do another one of these sessions sometime on how the Hoverfly Recording Scheme handles data. Obviously, I'd need to talk to Roger about that, because it has to be a joint session, because he deals with the species verification side of it. But I mean, it probably would be ideally sometime next autumn or whatever, when we've got some incoming data we can use as examples. But it's something that hopefully people will be interested in, but it would be nice to actually have documented and there so that people can see it, I think. So I don't know what the feeling is as whether they would be interested in that.
I would have said that there would be terrific interest. One of the things that the Facebook page, I suspect many members will feel, is it's sort of a black box.
Yeah.
And it would be very interesting to see what goes on behind the scenes. So yes, we ought to do that. And I mean, Alan, you're in a position to perhaps comment. You're as an observer from outside.
Yeah, I would have thought there'd be lots of people that would want to see what happens with the data and how it gets from A to B to C to D. I can kind of guess from the bits I see, but I think I think you'd get more people than we've had for these set of talks.
Yeah,
I would have thought so. Yeah.
Right.
Yeah, this is quite
some-
specialist.
Something to
Yes.
think about anyway, but maybe something in future.
Right. Well, that's another one to go for then, isn't it? We can.
Yeah,
we
maybe
can plug that.
like say a late end of years thing, because as the season winds down.
Yeah.
Yeah.
As we start getting data in, that would be the time to do it, I think.
Well, we've already got some, I'm afraid, Stuart.
Yeah. Oh, yeah. Yeah.
So we could do it at any time, but it would make sense to do something. I don't know, December, January time, something like that.
Yeah, that'd be, because it's hard to do anything else at that time of year as well, to come up with talks, because there's obviously aren't any hobbers around to inspire people to look at. So that'd be, that's a better time of year, I think, in terms of having something to talk about.
Okay. If there are any other sort of analytics or topics that people are particularly interested in, then certainly raise the suggestions and we'll see what we can do. But I mean, clearly it would be nice to have some of this documented, because we're not going to be here forever, and people are going to have to take it over. This is one way of actually making some of that stuff available.
Very much.
Could I ask how many people are using R on in this entomological community to look at data? Are the moths people doing similar things?
No idea, but certainly it is. If you look at the publication side of it, it's by far the most quoted statistical package in published papers. And I think,
in the general academic literature, I was thinking within the amateur community, as it were.
Yeah, I've got no idea, I'm afraid.
If you're going to be doing any sort of ecological or statistical modeling, R is, for better or for worse, the de facto language that people use.
Yeah.
It's not without its faults and foibles, but overall it does a very good job of being easy enough to pick up language, combined with being powerful and strong enough to handle things. It has a lot of pre-built packages which do a lot of the heavy lifting.
Yeah. Yeah, well, both my sons are physicists, and both of them use Python for most stuff, and R is regarded as too slow for the number crunching. But I think you're right. Within biology, ecology, medicine, finance, feels like that. It is all pervading, really.
But to your wider point, Phil, as a member of a number of groups, I don't see any sign of anyone doing what Stuart does in any other groups. I think it ends up feeding into academic studies more than anything else.
Yeah. Well, it's I mean, my experience was I've been a certain amount of data processing, I suppose, and analysis on Microsoft Excel, but I think I was getting towards the limits of that and needed a proper programming language that I used to have 30 years ago or more. And when I sort of saw R, actually, it does remind me of what it's like 50 years ago with the console and the blinking cursor. You type in things and it went to your local, I can't remember what it's called, VAX computer, and you got some results, because it's fairly simple calculations rather than all these high-powered packages, but nevertheless, it's there's all sorts of things I've been thinking of doing that I can now have a go at doing.
Yeah, I think Phil, the general point would be that probably 95% of recording schemes and contributors or associates of recording schemes are not actually interested in that side of biology. The Hoverfly scheme is essentially a bit of an outlier because of Stuart. I mean, I'd have to say that if it was me running the scheme without Stuart, there wouldn't be any modelling. I'd be a techno numpty like everyone else. Well, I am a techno numpty.
You'd be using spreadsheets.
Sorry?
You'd be using spreadsheets.
I probably would.
Oh, we can't be having recording schemes using spreadsheets. I'm sorry. That's
That's exactly why Stuart says I'd be using spreadsheets.
Yeah, I suspect there's a lot of them that are.
But yeah, well, that's what I've been trying to say to the other recording schemes is, what are we actually hoping to get out of this data, and are there tools we can start exploring the options? And
I think the problem that you've got actually, Phil, is that for the vast majority of recording schemes, the volume of data each year is tiny. I mean, we're now getting somewhere between 100 and 120,000 records a year. How many Anthemide records would you get? Maybe 1,000, 1,500?
Oh yeah, well, it's a 30,000 total already, but this is why I'm sort of, not looking at a microscope, but looking at a much broad brush scale. And these, I think there is a lot more mileage in these diversity patterns if you look across a wide range of species. And it seems to be something that's hasn't actually really been looked at Diptera, even though there was a lot of work on it 20 and more years ago in the general field of understanding these species abundance distributions. So, and now see where it goes.
It's one of the questions that I find quite fascinating. What is the real pattern of hoverfly species richness across Britain? And it's not that easy because it's so tied up to recording effort.
Yeah, well, I think that's why I'm hoping to get some kind of these chow et al. Methods to extrapolating to total species richness, knowing the amount of recording. We can, and if there is a common, well, I'm looking the other way around. If it follows sort of basic ecological pattern that works for hoverflies, if you understand it from that, does that apply to the less recorded areas where we're not seeing a big coverage in detail, but with the Apomida, we've got a lot of quite good coverage in particular areas where people have done like Stephen Falk in Warwickshire, the notebooks we've just decoded, and that's fits in with the pattern of the species area relation almost perfectly, as well as the log series distribution. So I'm trying to collect more data to test these ideas.
The thing to bear in mind, though, Phil, is that hoverflies are a little bit unusual because of the range of biologies of their larvae.
Well, are they? I mean, it's our Anthemaiad statless varied, and in extending this to, I've looked at across train flies, Impididae, hoverflies, calyptrates, and a range of other larger Diptera. So I think we're getting the same range of ecological functions as there are within hoverflies. So yeah, I think it's partly there's one explanation of some of these patterns is that it's different sort of maximum entropy. They've got so many interactions, so many different species doing different things that you get the most kind of possible disordered distribution within certain constraints. But I think that's a completely different application of maximum entropy from what Stuart's been talking about. Anyhow, it's a fascinating exploration, which we'll see how it goes.
Okay, so should I stop recording now?
Yeah, I think you probably ought to, Stuart.
Okay.